Hi everyone, I'm Rémi DANGLA. I'm the CEO of Stilla Technologies, a company based in France and in the US, focusing on digital PCR technologies applied to life sciences diagnostics. And today I'll be explaining how digital PCR can be usefully used for the detection of COVID-19 and the SARS-CoV-2 virus. So first of all, I'd like to thank the organizers of the Laboratory Automation and Informatics event hosted by LabRoots for giving me this invitation to present digital PCR and how it can be applied to the detection and diagnostics of SARS-CoV-2 and COVID-19. This is a great opportunity. Obviously, this is very much critical aspect of what needs to be done right now and the current situation of the epidemic. And so I hope that you can all learn today about digital PCR, its advantages over our real-time PCR, and again, learn how it can be applied for this critical application of the detection and diagnostics of SARS-CoV-2. So before I go into details of the presentation, I want to explain again what is really the need of diagnostics for COVID-19. So as of today, there are about 4 to 5 billion of reported cases of COVID-19 around the world. This is a worldwide epidemic that is still growing in exceptional proportions, and we have about 300,000 deaths accounted for as of today. And that again is a growing and rapidly growing number. So it is obviously an exceptional epidemic that needs to be treated with exceptional measures. And there's a common understanding right now that a critical solution for us to get out of this epidemic situation is going to be testing again. But when it comes down to testing, you essentially have two types of tests. You have the molecular tests that can be used for diagnostics, and you have the serological test that really tests for immunity. And for the future of the control of the epidemic, it's really the diagnostics tests which are going to matter, because diagnostics tests are able to diagnose patients who actively have the disease and therefore diagnose patients that present a risk to contaminate others, and those individuals really need to be identified, and we need to take measures to make sure that they don't infect others. Serological tests are obviously also important, but they only tell us who has been infected before and who potentially has immunity. But that will not allow us to prevent the spread of the disease further if we only do serological tests. So really the key, in my opinion, is going to be a diagnostics test. And as of today, and it's most likely going to remain as is in the future, diagnostics tests are mostly performed using what is standardly called RtQ Pcr or Rtbcr. So PCR with the retrotranscriptase step done in real time. And this RT-qPCR technology is now very common and used worldwide to perform millions of tests every day. And again, those tests really are targeted at patients and individuals who actively have the disease. And those tests allow us to detect the virus from a patient sample. The workflow of RT-PCR diagnostics I believe is now common to many people around and probably everyone in this audience, but it starts off with the sample collection, mostly done using nasal swabs. And then the swab is put into transport medium as a second step. And there's usually a transport step to get the sample into the clinical facility that will perform the test. And to perform the test, you have two main essential steps. You have the extraction of the viral RNA from the transport medium, the extraction step, and then you have the analysis by a real-time PCR. So it's a 4-step workflow, and there are out there solutions that have integrated some of those steps, combining the extraction with the analysis, but in general, this is the standard workflow. which again is performed routinely in millions of tests a day. But what we know as well is that there are definitely some limitations to RtQ Pcr testings. And there are multiple reports where those tests have not been able to correctly diagnose patients. So one limitation that is known is there have been frequent accounts of patients having pneumonia and having CT abnormalities that can be clearly seen on CT scans. So you can see such a CT scan on the right, where you see in the lungs of the patients clear evidence of liquid being present in the lungs. So the patients are obviously CT positive, but the results of the real-time PCR test has come out negative. And that obviously is a false negative test, and is usually confirmed either by doing a test by RT-PCR later on or by doing some sampling directly in the lung of those patients. And that has been reported in multiple journals today. Similarly, there are accounts saying that real-time qPCR has a sensitivity of 50 to 70%. 50% is probably a bit pessimistic, but definitely there is a lack of sensitivity, meaning that there are about 30% of patients of individuals who have the disease who would not be detected by real-time PCR. And if you go back to the workflow I presented before, this could come from different origins. One, the sampling could not have been performed correctly. If you don't do the sampling correctly, if you don't actually use the swab correctly, you're not going to collect the virus and you're not going to be able to detect it. Also, it's possible that the real-time PCR analysis is not sensitive enough in itself. So it's missing some virus particles, some virus genomes in the sample that is being analyzed. And so in general, there's clearly a need to develop more sensitive diagnostics workflow in order to address this challenge of being able to reliably detect and diagnose patients with COVID-19 and carrying SARS-CoV-2 virus. And so at Stilla, what we decided to investigate is whether digital PCR could be a solution to address this challenge. So before I explain how we have addressed this question, I'll explain a bit how digital PCR works in itself. So digital PCR, for those of you who are not familiar with the technology, is essentially the third generation of PCR technologies. So the first PCR technology is endpoint PCR that is done very routinely in labs and was invented back in the 80s and can be then analyzed using electrophoresis gels. So that's on the left panel. It's a very useful technology, but it's not quantitative. It's not very sensitive at detecting DNA or RNA, nucleic acids. Then later on came the second generation quantitative PCR or real-time PCR, and I guess the best name for it is real-time PCR. So what real-time PCR brought to the table is the ability to detect an increase in the fluorescence of a sample as it undergoes thermocycling and DNA replication. And if you detect this increase in fluorescence, it means that there is presence of the virus that you're looking for in the sample. So this is a great technology. This is now the workhorse in all labs, but it has some issues that I just mentioned before, in particular sensitivity. It's also not an absolute quantification tool, so meaning you don't really know how many material you started out with. You can know whether or not you had more or less to another reference sample, but it's not absolute detection and quantification. And so later on came digital PCR, and digital PCR third generation really addresses the key issues of quantitative PCR. Essentially, it's an absolute quantification tool, so it really counts individual DNA, RNA molecules one by one. You don't need any standard curve. You're really looking at individual molecules and detecting those reliably. It provides increased sensitivity, so you can detect those nucleic acids with single molecule sensitivity. and you have high precision. You can measure the quantity with only a few % of uncertainty. So it's obviously a great technology, and again, focusing on the differences between RT-qPCR and digital PCR, the workflow would essentially be the same. If you think again about the workflow I presented above, you would still, with digital PCR, start from an extracted sample. So you would collect, transport, extract, and instead of analyze the sample using RtQ-PCR, you analyze the sample using another instrument, which is performing digital PCR. Similar chemistry, similar assays. So from a workflow standpoint, very comparable, so it can be easily implemented in labs that are already doing real-time qPCR. But real-time qPCR, as I said, is mostly for detection of DNA or RNA, whereas digital PCR brings to the table increased sensitivity, absolute quantification, and also, and that's I think is interesting in the current context of COVID-19 detection and diagnostics, it has low variability between samples, between users, and between labs, and it's robust again against inhibitors. So it can essentially be implemented in very similar ways anywhere you would perform the assay, giving similar results across the globe, which is interesting when you want to compare performance of assays here and there. But what has to be said again is that right now RT-PCR is the workhorse technology and is the COVID-19 diagnostic standard. And digital PCR still remains a research tool and is only today transitioning to diagnostics and mostly as a confirmatory tool for COVID-19 in particular. The hope we have at Stilla is that by using digital PCR, we could indeed have a valuable asset in the battle against COVID-19 by increasing sensitivity. And by increasing sensitivity, basically, we could diagnose patients who come in either late, early or late into the testing facilities. If you see this middle graph here, at the moment, real-time PCR is only able to diagnose patients who have significant amounts of virus from the samples collected. And this occurs at the peak of the infection. But we know that patients can already transmit the disease early on when they are asymptomatic. And so it's interesting to increase the scope of diagnostics to the left of this curve early on in disease. And also what's happening actually quite frequently in hospitals is that patients unfortunately come in a bit late, already having severe symptoms, but at a point in the disease where it's not the virus that's causing the symptoms anymore, it's the immunoresponse of the patient that's causing the symptoms. And in those cases, again, it's hard to detect the virus from nasal swabs using RT-PCR, and so digital PCR could also expand the realm of diagnostics to the right of this curve. And if we basically increase our ability to diagnose patients, we can really understand better who has been infected by the disease and who is not, and then target how we want to treat patients accordingly. So how digital PCR achieves higher sensitivity, higher precision? It's by using a process, a workflow, which amounts to the following. So you start off with your sample that you want to analyze that contains multiple amounts of DNA or RNA molecules, so various amounts of DNA molecules. Instead of analyzing the sample in bulk, you start off by partitioning the samples into tens of thousands of subpartitions. So you cut up your sample into microvolumes, and by doing so, you isolate the DNA or RNA molecules in different compartments. So you initially have a complex sample on the left, and then when you look at a single partition, it's a very simple subsample where you only have one or two DNA molecules or RNA molecules. So you basically have simplified your sample. And then you're going to perform PCR in all those partitions, but each PCR is going to be essentially very clean because you're going to start by your PCR reaction with a single molecule, and you don't have cross reactions happening, you don't have noise in your PCR reactions. And so you get very clean PCR amplifications. And this PCR amplification is then combined with some dyes that will color the droplets or the partitions that contain the DNA or RNA molecules of interest in some color, so blue in this case. So at the end of PCR, you have a number of partitions which are colored and some who are not colored. Those who are colored become the positive partitions, those who are not are the negatives. And then by just counting the number of positives and negatives and applying some statistics, you can go back and calculate very precisely the amount of DNA you had in your sample. So in this example, you can get only 2 or 5% precision or uncertainty, which is really remarkable. But also think about the fact that if you had a single partition becoming positive, this would mean that you had a single DNA molecule in your sample, and you would really reliably detect the single molecule. So you can have single molecule sensitivity. So the workflow that I'll explain a bit more in detail is the Crystal Digital PCR workflow that we have developed at Stilla Technologies, where we do this 3-step workflow partitioning PCR and analysis in a format which we call droplet crystals. So we partition the sample into tens of thousands of micro droplets, and then those micro droplets self-assemble into a large 2D array that we call a droplet crystal. And then we do the amplification step, and we do the readouts using 3 fluorescence channels. So we do the readout using a blue, a green, and a red fluorescence readout. And this allows us to detect 3 different DNA or RNA targets and reliably quantify those 3 different DNA or RNA targets in our sample. So this is our unique workflow to enable multiplexing, precision, and sensitivity in digital PCR. To perform the workflow, we have developed and we implement in the lab to automate this workflow. We have our NICA system. So the NICA system is built around a key consumable, our Sapphire chip. So this is a microfluidic consumable that really integrates and automates the whole digital PCR workflow. And this is the key aspect in digital PCR. How do you automate the partitioning step, the amplification step, and the readouts when you have to process tens of thousands of partitions in parallel? So this automation is performed using microfluidics, and in our case, using our Sapphire chip. So the Sapphire chip is then processed using 2 different instruments, the NICA Geode and the Prism 3 instrument. And at the end of the workflow, also very important to enable our users and to enable those who will be performing the test, we have our intuitive Crystal Miner software that analyzes the data and gives results out to our users in the numbers of DNA, RNA molecules present in their sample. So our solution really has 3 main advantages when it comes to performing high precision genetic analysis. It's easy to use, as you'll see. It's a very simple process, and it's all integrated in a single consumable. It's also a fast process. It's the same time to result as standard RT-PCR, so about 2.5 hours to get your results out of the Sapphire chips. And we also have this ability, this unique ability to detect RNA, DNA targets in 3 colors, so you can get more information out of your samples. So as I said, the heart of the product is the Sapphire chip. So if you look at the backside of the Sapphire chip, this is where the automation takes place. It builds upon our proprietary and patented partitioning technology, which allows us to take about 25 microliters of PCR mix that is put in the chip and transform that into an array of 25,000 to 30,000 droplets in each of the 4 chambers of the chip. So each chip is capable of processing 4 samples in parallel. And we can reliably make those 25 to 30,000 droplets in our samples using this microfluidic technology, and then process the whole chip, the whole samples in the workflow in the chip. Ease of use for us is key. We have a workflow which can be performed by any lab technician that is accustomed to doing PCR, RT-PCR. So to really prepare the chip, you only have to take the chip, remove the white caps, and then pipette 25 microliters of PCR mix into the inlet ports. The chemistries that are being used are chemistries that are routinely found in PCR labs. So we have a specific master mix that needs to be used that we provide from Quanto Biosciences, but then it's compatible with any type of probes, Tagman probes, or even evergreen intercalating dyes to perform a variety of assays, PCR assays that are being done in labs that would adopt the technology. So once you've loaded the chip, you transfer the chip into the geode, and the geode will inject the PCR mix into the chip. And when it does the injection of the PCR mix into the chip, it spontaneously breaks down the PCR mix in those droplet crystals of 30,000 droplets per sample. And immediately after, it performs the PCR amplification. So the partitioning and amplification steps take about 2 hours and 10 minutes. And once this step is performed, the chips are transferred to the second instrument, so the PRISM 3 instrument, which takes high-resolution images of the droplet crystals using 3 different fluorescence channels. So a blue channel that can detect typically FAM dyes, a green channel that can detect VIG, HEX, ROX, Cy3 dyes. So a broad range of dyes can be used for this channel, and then a red channel that can detect Cy5 or Cy5.5 dyes. Sorry. The readout is fast, so it's less than a minute per sample. So basically, in about 2 hours and 15 minutes, you have the results for your 12 samples that you would have processed in the geode. And the last step that needs to be automated is the image analysis, and we do so with our Crystal Miner software, which builds upon AI tools to do image analysis and extract the fluorescence of all the droplets that you have in your droplet crystals. So we process tens of thousands of droplets, image droplets, and we process them through our neural network software to make sure that we can reliably detect those droplets, exclude artifacts, measure the fluorescence, and basically give out results which are, one, reliable, but also can be intuitively analyzed by our users to classify the droplets as positive for the blue channel, green channel, or red channel. So it's really a powerful software, which also provides quality control, trustworthy quality control. For example, in terms of quality control, something that's quite unique with the NICA system is that you can actually look at the droplet crystals at the images of those droplet crystals, and you can overlay on top of the images which droplets are positive for one or the other of your targets. And this is a reliable tool for users who want to understand really what's happening in their samples and look at the quality of the results that they're getting. So in summary, with this Crystal Digital PCR workflow and with the NICA system, we have enabled the automation of the digital PCR workflow in a single consumable with 3-channel fluorescence detection, and that takes place in about 2 hours and 30 minutes with only about 5 minutes of hands-on time. So really an automated workflow that can be simply implemented in the labs. And so this digital PCR workflow has already been implemented for a variety of applications. So the dominating applications, I would say, prior to this COVID-19 epidemic was oncology, but you had applications for environmental science, for food testing, for infectious disease, any domain of science where you need to do genetics, molecular testing, has potential uses for digital PCR, and digital PCR is really becoming the new standard for precision genetic analysis. For those who are interested in learning more about digital PCR in itself, I can point you to a website called GenePy, which we have built together with some collaborators as really an educational portal for people who want to learn how digital PCR performs, learn about the math behind the statistics and how it can be applied to different cases like rare event mutation detections, like copy number variation quantification, or to perform more complex assays like drop-off assays. So a really good tool to learn more about digital PCR in general, not just on the NICA system, but in general, how digital PCR can be useful for genetics. So let me now turn to how digital PCR again can be used in the context of COVID-19 and our battle against the epidemic. So as I said, something that we investigated at Stilla is whether or not digital PCR could be used to increase sensitivity and reduce the false negative rates that have been observed by real-time PCR. So to do so, the first thing that we need is an assay that can be used to detect the SARS-CoV-2 virus. And so we have developed an assay to do exactly that with our partners Apex Bio, who are based in Suzhou in China. So we have worked with Apex Bio Suzhou very early on in the outbreak of the epidemic. We were luckily in contact with our partners there who were first to witness the outbreak. And so as of starting even in January, we started working on the development of this assay, which is currently the only triplex assay for the detection of SARS-CoV-2. So the assay is set up as following. So we have detection on the FAM channel of the ORF1ab gene of the SARS-CoV-2 virus. On the green channel, we detect the nucleoprotein N of the viral genome. And in the Cy5 channel, we have a human housekeeping gene that's being detected as an internal endogene control, which is also very important to make sure that when you get a negative result, it's a true negative and not a malfunction of the instrument of the assay itself. So this assay is available to any user who wants to implement the NICA system in digital PCR for SARS-CoV-2 detection, and it's available worldwide. And before I go into the presentation of some of the results we've obtained with this assay, I'll talk a bit about the design of the assay itself and how you can use the multiplexing capability of the NICA system to have increased sensitivity. And so here what's shown is a picture of the structure of the viral genome. So what's interesting with SARS-CoV-2 is, it's a very long genome for a virus. It's almost 30 kb long. And so we have chosen to have two different targets in our assay that are spread apart one from another in the genome. So one of our targets is ORF1ab gene. So it's located to the left of the genome. And the other is on the N gene. So those two targets are separated from one another with a distance of more than 10,000 base pairs. So they are very spread apart. And what's interesting here is that in practice, viral RNA, once it's analyzed in digital PCR or real-time PCR, the viral RNA is quite highly fragmented. This fragmentation of the genome occurs probably already at collection. It most likely occurs during transport, where the virus itself is lysed. And then it also occurs quite extensively in the extraction process. So when you analyze the virus itself, you don't analyze 30 kb fragments. You only have fragments of a few hundred kbs. And so one genome actually becomes two different fragments at the very least detected on our system. If you have one genome in your sample, you would get one green droplet and you would get one red droplet. So you get two events per genome. And so basically, by looking at the two different channels, you basically double the probability of detecting one genome by looking at those two different fragments. And that's something that's quite unique with digital PCRs, because you have this single molecule detection capability on each channel. By looking at the two channels simultaneously on the same assay, you really can increase sensitivity when you detect a single droplet in either of the channels. It tells you that you had one genome, and you reliably detect one genome of the virus in your sample. So that is something to keep in mind when you design an assay in digital PCR, and you think about whether or not the DNA is fragmented or not. You can cleverly use the assay design to increase sensitivity, for example. And this is what we've done. So we've checked what the sensitivity of our assay was by running some spiked experiments with positive controls of ARF1AB and nucleoprotein and sequences. And something that's expected in digital PCR, you have very good linearity of your dilutions, so you can really precisely quantify your targets across a broad range of viral concentrations. And we have found that we have a limit of detection that is about 0.6 copies per microliter and 0.9 for each of the two genes. And again, those are concentrations where you have 95% probability of detecting the virus. And that is lower than what has been found in a real-time PCR, at least twofold, if not fivefold lower than what is custom with real-time PCR assays. And again, there are approaches which allows us also to combine those results and increase the sensitivity by looking at the results together and not separately. Forgot to mention, obviously, specificity is important as well, and we have observed no false positives in negative samples, and specificity is also verified in this assay. So the first results I want to show on patient samples is a study that we performed in collaborations with a hospital in France, CHU de Rennes. And this study was really targeted at looking at samples where the patient came in, the patient had a CT scan which showed abnormalities. So the patient had pneumonia-like symptoms, they had CT abnormalities, but then when they performed PCR on nasal swabs after extraction, the PCR results came out negative. And so the hospitals are really having issues now with such samples and such individuals, because in the hospitals, they don't know if they should put such patients in COVID positive facilities or COVID negative facilities. And yes, today COVID-19 is a major contribution to pneumonia right now, but it's not the only reason why people have pneumonia. And you don't want to put someone that has pneumonia but not COVID-19 next to people who have COVID-19. That can only make things worse. So we wanted to check whether or not the NICA system could be helpful here. And so we used our NICA system together with the COVID-19 kit. And so the early data that we can share, and that's data we only got as of last week, so this is very recent results. We took 10 of such patients that came in the hospital, and we processed them in qPCR first. They were negative, and then we reprocessed them in digital PCR. And out of those 10 patients that were negative in qPCR, 4 came out to be clearly positive in digital PCR. So that's a significant improvement in terms of diagnostics. For the 6 others, they are negative in digital PCR. They could still have very low amounts of virus that was undetectable, but it's likely that those are actually negative patients, and it's probably makes more sense to treat those positive digital PCR patients positives in COVID positive facilities in the hospital. So it's a 40% increase in terms of sensitivity on those specific qPCR negative patients. And that's a significant result obtained here, showing how digital PCR can be used as a confirmatory tool for diagnostics of critical patients as those. A similar study that can be shared today is one that we performed at the IHU Marseille. Again, it's a hospital in France. And there, the goal of the study was to look at qPCR results, which are basically dubious, doubtful. So when you have a qPCR results, you typically look at the CT value that comes out. So at which cycle do you get a fluorescence readout for your sample? And typically, when the sample has a lot of virus, the CT value is low. You find out fluorescence values early on in the amplification step, so the CT is below 35. But when you have low amount of virus, you can have amplification at very late stages when the CT is then above 35. But it's unclear if when you have a CT that's above 35, whether this is a true positive result or if it's positive because of some polymerase error or some amplification that's non-specific to the SARS-CoV-2 virus. So we looked at a set of samples. We looked at 15 samples that were negative by qPCR, 12 that were clearly positive with CT values below 35, and 18 who had doubtful results. First interesting result is that amongst the negative, we had one recall, a sample that was clearly positive by digital PCR. Again, it's likely to be due to the increased sensitivity of digital PCR compared to real-time PCR. Out of the 12 positive samples, we could see that we could reliably detect the virus by digital PCR, and also we found a good correlation between the CT value and the quantity of virus that's detected by digital PCR. The lower the CT value, the higher the concentration measured by digital PCR. The correlation is not full because here the qPCR assay is not standardized, and so it's hard to really say and compare qPCR results one to another. Something I pointed out to before, without any standard curve, you can't really compare patient A to patient B, which could have been processed differently, which could have different contents of inhibitors, etc. But at least there is a good correlation as expected. Now looking at the 18 doubtful results in qPCR, those had CT values that ranged from 34 to about 40 in terms of CT values, and we processed them by digital PCR. And the key results is that we actually reliably detected the virus in 11 out of those 18 samples. 7 only out of 18 came out negative. And so here again, we have more than half of those doubtful results, which become clearly positive by digital PCR. And we could then use this confirmatory tool in terms of patient management in hospitals to make sure that patients who have high CT values but who are digital PCR positive can be put in COVID-19 facilities in hospitals, and those who are negative on both can be treated separately with less risk of infecting others. So those are really two studies, preliminary at this point in time, but who interestingly point out clearly advantages of using digital PCR compared to qPCR. What has to be said is that this approach is now routinely used in China in the Center for Disease Control, which are using digital PCR to confirm doubtful results on several hundreds of samples. And this is something that we hope we can also implement in Europe and in the US with our approach. So this was an illustration of how digital PCR can be used to COVID-19 detection and quantification, but digital PCR, as I said, can be used for a number of other challenges in the realm of genetic analysis and molecular diagnostics. So we have a number of application notes that can be found on our website. And those results and this approach can also be found in the publication we have on our website, specifically dedicated to COVID-19 detection. So with this, I conclude my presentation. So I really want to thank the doctors and physicians who have helped us analyze samples, patient samples at Institut CHU de Rennes and at the IHU de Marseille. So Professor Michael Drancourt and Dr. Amar Bouham. So a big thank you to them. And again, thank you also to the organizers of the conferences for giving us the opportunity to present those results on how digital PCR can be a helpful tool in the fight against COVID-19. Thanks for your attention.